Hello, and welcome to Science with Faith. Today we are going to be learning about protein synthesis. Protein synthesis starts with your DNA, so let's give a little pre-lesson on DNA so the rest of the lesson makes sense. The DNA in your cells contain crucial information that makes you, you. DNA stands for deoxyribonucleic acid. It has a double helix structure and is found tightly wound in the nucleus of all of your cells. DNA is made up of a deoxyribose, or sugar, and phosphate backbone. The rungs on the DNA are made of adenine, thymine, guanine, and cytosine. Adenine always pairs with thymine, and guanine always pairs with cytosine. The pairing in the DNA sequence is known as your genetic code. All your cells have the same genetic code. What differs is the types of protein the cells produce. The DNA is replicated in the cell cycle. Whether it is an asexual or sexual body cell, that cell cycle will differ. DNA is unwound by the catalyst enzyme known as a helicase. The helicase is able to unwind the DNA strand in half by dissolving the hydrogen bonds. The catalyst, DNA polymerase, helps the process move smoothly. The single DNA strands are paired up with free nucleotides. The deoxyribose and phosphate on the nucleotides form the new backbone of the DNA. Now that you know a little more about DNA and how it's copied, we can move on to how DNA becomes a protein. First of all, what is protein synthesis? It is a process that allows body cells to produce specific kinds of protein by reading a code. There are essentially two steps to protein synthesis: transcription and translation. Transcription starts off with DNA. A section of the DNA with the needed protein codes are copied. The DNA is unwound by the helicase. One side of the now unwound DNA is now used as a template to create RNA, or ribonucleic acid. RNA is a single strand and contains adenine, guanine, cytosine, but thymine is replaced with uracil. RNA can create mRNA, tRNA, or rRNA, otherwise known as messenger RNA, transfer RNA, and ribosomal RNA. A catalyst enzyme called the RNA polymerase lines up free nucleotides in the cell with the DNA template strand. All the genetic information is now copied, and mRNA has been created. Each strand of mRNA contains the code for a single protein. When transcription is finished, the mRNA is let go, and the DNA gets wound into the double helix again. The next step is translation. This starts when the mRNA leaves the nucleus and enters the cytoplasm. While the mRNA is traveling through the cytoplasm, it will encounter ribosomes. There are larger and smaller ribosomal subunits. During translation, the ribosomal subunits come together. Ribosomes are the particles responsible for protein synthesis. Ribosomes have different spots for mRNA, tRNA, and rRNA. Each strand of mRNA has a stop and start codon for what codes need to be read. When the mRNA finds its place in the ribosome, tRNA comes into place. tRNA is categorized by different amino acids, and they bind to the ribosome in the order given by the mRNA code. tRNA has the shape of a clover leaf. At the tail of the tRNA, it has a part that binds to amino acids. At the head of the tRNA, it has just 3 nucleotides that create an anticodon. A chain of mRNA enters the ribosome and is met with tRNA that complements their genetic code. That creates a peptide bond, and after a while creates an amino acid chain. The chain will stop when the stop codon is read. The amino acids are the building blocks for the structure of a protein. When the amino acid chain is long enough and contorts properly, it creates a protein. That is how your body creates proteins. Pretty cool, huh? Thanks for watching.